Way. Welcome to the urinary system. Tonight's lecture and recording is going to be about mainly the function of the nephron, which is the basic functional unit of the kidney. We're going to go through that, and then control of water balance, which is urine concentration, is another word for that. And then finally we're going to do some acid-base balance questions, because for the number of pages that's there that you're supposed to study, there seems to be an inordinate number of quiz questions and exam questions in the banks. And so you're likely going to see those even though there's only a few pages. And so I do want to spend time, because students most of the time miss most of those questions. And I think it's just because they think, oh, it's like 4 pages, 5 pages, no worries, I've got this. And then they see lots of questions on it. So once again, just wanted to reiterate that the nephron, this thing right here, is the functional unit of the kidney. And this is where we're going to spend most of our time. There's approximately 100 million nephrons. And they are not something that we regenerate. These are not cells, at least the nephron tubule portion, that we see regenerate easily or at all, depending on which cells we're actually talking about. And so when these are damaged, this is why we have one of these organs that needs to be replaced, or at least one of the two functioning, in order for someone to function normally. It's not something that just gets better over time or can heal itself in that sense. And so it's important we pay attention and care for our kidneys, make sure we're doing those things necessary. And when there is kidney damage or kidney disease or even passing kidney stones, that's watched really carefully so that there isn't extra damage. All right, but yeah, wonderful system in place, again, with our Father in heaven and the body, where there's a redundancy. So you have an extra kidney that can really compensate for the one if the one's damaged. And fortunately is something that we can receive a transplant for, and that can function, fairly well on its own. So if you have to have one. Anyway, still, you see that all the time, that the body's really good at being able to heal itself with most cases. And if it can't heal itself, there's often some other backup plan before it just gets to the point where it's too much. And of course, external things like environment and genetics and other things, contribute to our fallibilities of our bodies. But when you look at it as a creation, I think it's pretty awesome, the things that it can do on its own and how it does kind of function the way it does function. Anyway, so moving on. So today we're going to talk about, essentially, just like the digestive system, where in the recorded lecture that I had from last semester, if you just take your meal and go through the digestive tract, that's probably the best way to learn all about the digestive system and what happens and where it happens and what happens to what we eat and that type of thing. We're going to do the same thing with the kidney. We're going to start in the proximal convoluted tubule, which happens to be over here, and take the filtrate, which comes through the Bowman capsule and the glomerulus, and take it through the nephron to the collecting deck, and just kind of talk about what happens in each section is probably the best way to talk about its main function, which is to regulate body fluid composition. And that composition entails, glucose and electrolytes and toxins and water and all of that. So your kidneys are really essential for proper function, because they're removing toxins, removing excess electrolytes, saving electrolytes that you may need, and subsequently causing water to leave or keeping water depending on the need of the body at the time. And so, a really essential organ and essential functions. So let me grab the first part, which is the proximal convoluted tubule. I don't really like this picture, so I'm looking in your book for a better picture. Oh, let's try that. I'm going to go to process figure 2616, and that's on page 969. And this is the urine concentrating mechanism thing, but it has the nephron kind of spread out, and that's why I like it. And I'll refer to other figures, but this one is one that is easy to discuss, the nephron. Oh, that one too. Let's look at this one really quick. This is figure 26.4. This kind of shows its cross-section of the, let's see, cross-section that you can see where the cortex is and how a portion of the nephron's in the cortex and a portion's in the medulla. And I think that's important to understand because there are some tissue differences in between the medulla and the cortex that allow for some of the things that we're going to see here in the loop of Henle. And anyway, so here's a cross-section, nice pretty diagram. Once again, this is 26.4. You should know that one too. Okay, let's go back to 2616. All right. Let's see. Maybe, maybe not. Oh, they're backwards this time. Okay, there it is. Alrighty, so like I said, I want to spend time mainly so I can just talk about the different sections. Proximal convoluted tubule, we're going to come down the descending loop of Henle, the loop of Henle, the ascending loop of Henle, the distal convoluted tubule, and then finally in the collecting duct. So we'll address this a little bit later, but I'm just going to go through what happens with the filtrate in these sections. Now the filtrate, you should know, is the glomerulus in the Bowman's capsule is kind of like a sieve, so you're not going to get any red cells or white cells. They're too big to go through this sieve of the filtrate. So the filtrate's mainly water, small molecules like glucose come through, drugs, hormones, that type of thing. A lot of electrolytes, sodium and chloride and potassium and calcium, those all come through, and of course water. And so the filtrate is essentially that collection. You can think of it kind of like serum. So you don't have plasma proteins either in the filtrate. They're too big. So just the small stuff comes through. Very, like I said, very similar in what we've talked about before as serum. So that comes through. And as it comes through into the proximal convoluted tubule, the majority gets reabsorbed right back into the interstitial fluid and then subsequently gets back into the blood. And there is a lovely table, which, as you know, McGraw-Hill doesn't let me download tables, but let me find it really quickly. But it tells you, let's go to table 26.3 on page 960, gives you, in the proximal convoluted tubule, all the things that are the major solutes that get reabsorbed, and that includes amino acids and glucose and fructose and lactate and citrate and all those electrolytes. Now, glucose is, in a normal, healthy person, 100% reabsorbed in the proximal convoluted tubule. So it goes into the filtrate, goes right back, ultimately into the blood. It's a major energy source, and your body's going to want to keep pretty much everything it can for that. And so if you do see glucose in the urine, then you know there's a problem. And we talked about that a little bit in digestive and a little bit more in endocrinology section, but that's diabetes mellitus, or excess amounts of sugar in the urine. And that's what people mostly think of when they think about diabetes. But we're going to talk about diabetes insipidus tonight, which is excess of water in the urine. But anyway, so the large bulk of glucose and other nutrients like fructose and so forth will come out in the proximal convoluted tubule. We see sodium and chloride and potassium and calcium. All those come out as well. And when I say come out, I mean they leave the tubule, they leave the nephron and go into the interstitial fluid, and it's very closely closely mirrored by the blood supply to the nephron, the vasa recta, and it's just going to go right back into there. So you don't have to worry too much about that, but do know that those important things get reabsorbed right back in proximal convoluted tubule. As the filtrate comes down past here, you'll notice these numbers over here. They start to get bigger and bigger, and we're going to talk about this in a minute, but this is the osmolarity. Osmolarity, once again, is the concentration of solutes in a fluid. This fluid is water. So you're seeing that the fluid is becoming more concentrated with solutes. In other words, more water is leaving. Now there's a couple things. I mentioned one of these a couple weeks ago. Wherever sodium goes, water follows. So as sodium gets reabsorbed or gets removed from the filtrate, water is going to follow that. And water follows in a much bigger I don't want to say concentration, but in a much greater quantity, and so that's why you start seeing the increase of this osmolarity as water's just pumping out. As you can see up here, 65% of water gets immediately reabsorbed, but we're still going to reabsorb lots of water here in the descending loop of Henle. And part of that is because sodium is leaving, and so water follows. You can see it over here as well. They're showing sodium, but sodium leaves here too. And wherever sodium goes, calcium likes to follow also. Or not calcium, excuse me, chloride likes to follow also. So we're receiving back into our bloodstream the sodium, the chloride, the water, that type of thing. And that's pretty standard in the descending loop of Henle. Here at the loop of Henle, you'll see it's super concentrated. And that's one of these concentration mechanisms, Amelia, that you asked about. We have this, what we call a countercurrent, and it's also been referred to as a recirculating gradient. So what happens is we have the medulla here, which is really concentrated in solutes. So you're going to see that water, just due to the concentration in here, is going to actually leave. It's following sodium, which also helps it leave. And when it does that, you're going to get much more concentration coming in here. Well, the countercurrent is that some of the solutes are actually coming in, or diffusing into the loop of Henle because of the concentration gradient difference. So if you recall diffusion, it's from a high concentration to low concentration. And that's actually happening with a couple of the solutes themselves. You actually see that the solutes will come in to the tubule because of the concentration difference, but it's so concentrated here that water comes out, so you get this counter current, and so water will leave, some water will enter, solutes leave, such as sodium and chloride, but other solutes will enter, and so you get this once again recirculating gradient where in solutes and water leave, so you think that this would drop, it would be lower, but then solutes go in to make it higher. They do go into the vasa recta, and then they go back into the, which is part of our bloodstream, so to speak, and then they go back in. It's just so happens that there are properties about the vasa recta that don't allow that to keep coming back. So the solutes that are coming in are actually coming in from the interstitial fluid, and the water and the solutes that are leaving end up in the vasa recta, so it ends up in the blood flow. So that's why you get this really concentrated value down here at the loop. It just kind of goes in and out and in and out and in and out type of thing, and it kind of cycles through that way. And so you're seeing, like I said, not just sodium, but you see potassium, and you see chloride, and you see some of those other solutes that are actually leaving, but some are entering from the interstitial fluid. But when they leave, they go into, like you mentioned, the vasa recta, which has a different type of property that doesn't necessarily allow all of that same permeability. So it's not the same as just diffusing from the interstitial fluid. You may see some, because wherever sodium goes, water will follow. So water may still come out of the vasa recta if sodium is leaving, but you're not going to see the same quantities. And so you're going to see it becoming more and more concentrated as it gets closer to the loop. And once again, you see, they're still seeing a percentage of water, a pretty big percentage of water coming out. Well, properties change as we go into the ascending loop of Henle. Here, sodium and chloride are still being removed from the filtrate, but this area here, well, this area, bigger area, and the ascending loop of Henle and the distal convoluted tubule is actually impermeable to water. So as the solutes leave, the water concentration stays the same, and because of that, you start getting a lower osmolarity within the filtrate itself. Now, there are exceptions to that water, and that has to do with hormones, and we'll talk about that later. But under regular conditions, this tissue, this tubule area, is impermeable to water, so water can't leave even if it wants to. It can't. And so you see sodium and chloride and sometimes potassium will leave, but the water's stuck essentially, and so you once again get a very dilute urine right here. Okay, and you can see a 200 milliosmol type of thing is actually more dilute than your regular interstitial fluid. Same thing happens in the distal convoluted tubule. One of the other unique things about the distal convoluted tubule in this area is we may get secretions. Now, when I mention a secretion, that's talking about things like free hydrogen ion, toxins from the liver. So if your body, for instance, as we know, the liver breaks down drugs and alcohol, those basic units that it breaks it down will be secreted into the distal convoluted tubule and this portion here of the distal, well, of the ascending loop of Henle. By here it's the distal convoluted tubule. But secretion will occur here, and so you'll get that extra stuff coming in. May make it a little more concentrated, may not, you know. So here we're at 100, here we're at 300. Part of that is that water may or may not leave, and we'll talk more about that. But part of that's also due to secretion of other products. And then finally, they all come through the collecting duct. And you can see the collecting duct still shows some sodium chloride and some water being removed from the filtrate. And a lot of this right here is due to the needs of the body as far as blood pressure as a measurement of the water volume that we have, because we know that we store water in blood. And depending on the presence of hormones of aldosterone and ADH and ANP for that matter, and angiotensin too, will depend on whether or not we have a really concentrated urine or we have a really dilute urine. And so we'll talk more about that aspect in a little bit. But you still are seeing in the collecting duct some of these solutes leaving the filtrate itself. And so the filtrate, once again, can be very dilute or it can be very concentrated, and that is actually just dependent upon those hormones that we're going to talk about. All right, let's see. Let me show you this one. It gives you another little bit of difference, and this may have been the better one to show you initially, so sorry about that. Looking for 14. Sometimes they're a little too selective. Yeah, it doesn't look like I have 14, but 2614 kind of shows that filtrate concentration and its interaction with the vasorecta on page 967. So if you have questions about that, that is a good place to go. This is just showing the histological differences that you can see in here. So you can see that here, proximal convoluted tubule looks very different than the distal convoluted tubule. And these properties, these differences in how they are, will also help with the differences in whether or not water can be reabsorbed or not, and whether or not certain electrolytes are being actively transported or if they're being diffused. And so you see both methods of transportation for solutes, both active transport and simple diffusion, happening there. So let's see. Sorry, I don't know why McGraw-Hill is selective with their figures. I download all of the ones that they allow us to, but it doesn't look like that one that I mentioned was there. So let's see. Your book talks about different things like urea and other things. I think if you get the basic idea of what gets reabsorbed and where it gets reabsorbed, for the most part, you'll be okay. That fundamental figure we just had, 2616, the table that we talked about that shows the electrolytes and where they're being reabsorbed. And then, let's see. And then they don't have a really good one for that. So, all right. That's all anatomy. Let's go then to, let's see. Is it this one? Let's just look at this one. It's hard to see. They're so teeny. All right, so this is one of the first concentration mechanisms, hormonal mechanisms that your book talks about. And this figure is from page 970, and it's figure 2617. And we actually talked about this not long ago, 2 weeks ago. when we talked about water balance. So your kidney is very sensitive to changes in blood pressure at the site of the glomerulus. Whitney, that's a great question, and the large bulk of them will all be in the proximal convoluted tubule. And as you can see, there's a whole bunch. You don't see glucose ever after that because it's all reabsorbed. It's 100% reabsorbed. A lot of the phosphates all reabsorbed. The amino acids, all that stuff is proximal convoluted tubule. You can pretty much guarantee it's 80 to 100% of almost all solutes. And then the remaining are electrolytes that you're used to: sodium chloride, potassium, some calcium. All those are going to be in much smaller quantities. The figures I've seen are anywhere from 60 to 80% of everything that we'll ever reabsorb is reabsorbed right in the proximal convoluted tubule. So keep that in mind, with that meaning those other things like lactate and glucose and fructose, all that's 100% from proximal convoluted tubule. Okay? And it may say that in your chapter in a previous, not this version, but the version before, well, not the 9th, but the 8th edition, I think, did say what percentages they were. But we're more concerned that you know that they all get reabsorbed, that the large bulk get reabsorbed in proximal convoluted tubule. And then it's more of an electrolyte balance type of game, not counting the secretion that happens. Then, you know, we're mainly just working with those electrolytes themselves, but it's still in much smaller quantities than what it was in the proximal convoluted tubule. Okay, so this is the renin-angiotensin-aldosterone system, and I know we've talked about this at least twice, but it's always good to have a refresher. As I mentioned, the kidney is very sensitive to changes in blood pressure. So when there is a lower pressure in that little artery in the kidney, I don't know that it has a specific name, but one of the ones that comes in there or comes into with the glomerulus, if there's a lower baroreceptor firing, so to speak, so lower stretch receptors are not firing as frequently, then that is a signal for renin to be released from the kidneys. Now, most students forget that renin is not a hormone. They get that wrong a lot on the exam. But renin is an enzyme, which means it's just a catalyst. And what happens is it takes angiotensinogen, which is a plasma protein that's produced in the liver, and it changes it, or, well, it facilitates a change to angiotensin 1. Angiotensin 1 is a vasoconstrictor. It's not a very strong one. But as it goes through the lungs, this other enzyme, the converting enzyme, shaves a couple more amino acids off and becomes a very strong, very potent vasoconstrictor, angiotensin 2, which then, as it goes through and stimulates the adrenal cortex to release aldosterone. There's also a little side note that potassium can do the same thing. As far as if we have lots of potassium, that means we're probably losing lots of sodium. Sodium and potassium, the sodium-potassium pump functions, as you know, as one goes in, the other goes out. So as we're reabsorbing sodium, that means we secrete potassium. And so as we're doing that, if it senses large amounts of potassium, then we know that there's low amounts of sodium, which means too much water is being lost if we're not getting enough sodium. So either one, but angiotensin 2 will stimulate the adrenal cortex to release aldosterone. Aldosterone is a steroid hormone, so it actually has its effect inside the nucleus of the cell itself. And what it does, and we've always just talked about it in general terms in the past, that it increases sodium absorption, which then increases water absorption. But as you can see here, it increases transport proteins. And then you have a higher increase of active transport of sodium. So you're reabsorbing more sodium than usual. And you also have increased antiports. So here's the specifics for this week that you have to learn, is that aldosterone increases transport protein mechanism, so you have bigger, or more rather, not bigger, but more active transport channel protein gates and those types of things that are happening to increase sodium reabsorption. If we increase sodium reabsorption, we increase water reabsorption. If we increase water reabsorption, we increase our blood pressure because we store most of that in our blood. And so that's the specific mechanism of how aldosterone functions. We'll wait for Amelia to type. So then while she's typing, the opposite would occur if we have a higher pressure. We would have a lower release of renin, lower active angiotensin II, so lower amounts of aldosterone. Then we would not have as many transport channel proteins, so you wouldn't reabsorb as much sodium. Yes, well, it goes back into the, it goes back into, essentially into the blood. So it may go right into the interstitial fluid, but that figure that I wasn't able apparently to download kind of shows that the filtrate, it then goes back into the vasa recta and back into the blood. So while it may travel a little bit through the interstitial fluid, just by other mechanisms, it's going to go back into the blood. So when I talk about reabsorption, I'm really talking about reabsorption back into the blood. And that's what happens with all the solutes and the glucose and everything. They end up back in the blood through that mechanism, okay? And like I said, if you look on 967, it helps a little bit more, and unfortunately I don't have that picture. That picture's 967. Okay. And that it gives you a little bit better idea. If you go back into the anatomy too, it kind of shows it a little bit better too. If I can find one of them. Well, maybe not there. So if you look on page 20, on page 954, on figure 26.7, which was the first picture I showed you, you can see that the capillaries are all over, you know, the nephron, and that's where everything's going to get reabsorbed back into. So it gives you another picture on 954 of how that happens. It's very similar to how we view and how we talked, although we talked really briefly about lymphatic nodules and the lymph tissue and how it almost is like another circulatory system right next to it in certain locations. And the same thing we see in the nephron itself. You have that circulation system kind of going right through their capillary beds and other things so that they can get those solutes back. If you look on pages 962 and 963. Oh. Okay. Thanks, Siri. I don't know if you heard her start. Oh, I found something on 954. I don't know how she figured out I was talking to her. All right. On page 962 and 963, it shows the tubule and it shows then the blood vessel. They're right next to each other. And so it just goes, you know, through diffusion and transport and symports. They just go right in through. So hopefully that will help to kind of get that picture in your head. Okay. So renin, angiotensin, aldosterone system. Lots of questions about this because you've had it before. We expect you to be familiar with it. We've added one extra piece here. Since this part should be all review, this is the extra piece of what aldosterone's actually doing. And so that's kind of, I think, cool that we can tell you a little bit, give you a little bit more, and now give you the real specific picture and understanding of what it's actually doing. It's not just this like magic box thing. It's actually increasing those transport proteins and helping sodium reabsorption, which once again helps water reabsorption. Now the other mechanism, and then we have a picture for this too, it is a fundamental figure rather, and it is 2618, and it's on page 972, and this is ADH. Now ADH, oh, that's lungs. Where is it? That it. We're going to pull this one up and just see. Oh no, that's not it. Sorry. I used to have it open with larger icons so I could see the pictures better, but then I found I kept clicking on them and wondering why they weren't functioning, and it was because I was clicking on the thingy on the window, not on the actual open thing. Okay, so this is ADH. Now ADH is the posterior pituitary hormone that's made in the hypothalamus and stored in the posterior pituitary. Its excitation, or what stimulates ADH, is the change in osmolarity. And you only have to change a couple milliosmoles for ADH to be stimulated and actually secreted. What happens is, remember that location where I told you in the ascending loop of Henle and the distal convoluted tubule, that water is impermeable in that portion of the tubule, in that portion of the nephron. Water can't be reabsorbed. Well, ADH causes water to be reabsorbed there. So it's a really potent hormone in the sense it really helps. Direct reabsorption of water is what's happening here. And so if you look at, where's my little arrow? If you look at what happens is, since it's a pituitary hormone, or even a hypothalamic hormone, actually, since it's created in the hypothalamus, we know it's a peptide base or a protein base, and so it has its site of effect at the membrane. So it binds to the receptor, it activates cyclic AMP, which goes through this cascade reaction and causes these aquaporin molecules to open or to allow water to be reabsorbed, or normally, if these are shut, not going to happen. You're not going to see water leave. And so it actually increases that permeability by the second messenger system with the proteins in the cyclic AMP. So once again, you've already had ADH. We've already talked about what it does and why it's stimulated and what happens. Now you're getting one more piece of that step process of understanding that it binds at the cell membrane, activates G protein and creates cyclic AMP, which then allows these molecules to be open, allowing for reabsorption to occur. So it becomes a selectively permeable membrane versus an impermeable membrane when ADH is present and is binding to the receptor. And once again, that happens due to a small change in milliosmoles. So if you're getting a slight increase, a little more concentrated, that means you have less water, and so it's going to cause ADH to be released so that you have more water, so the osmolarity goes back to normal. What's considered normal anyway in your body. Okay. All right. If you don't have any ADH at all, this is how potent this hormone is. If you don't have any ADH at all, and you don't have any reabsorption of water, you have a very dilute urine. You have an excess of water in your urine, and we call that diabetes insipidus. And there are a few questions, especially since you have digestion on this last exam as well, that we'll try to see if you really know the difference between diabetes mellitus and diabetes insipidus. So do take the time to read about diabetes insipidus on page 971 and the difference to diabetes mellitus, which it actually mentions also on page 971. That is kind of a critical understanding that diabetes is not, what everyone thinks it is, even though we talk about it in that term, because diabetes insipidus is really quite rare. But what that term means, if you've had medical terminology, then you kind of know that, we use words like that to describe something, and that's the second word actually that makes it more specific. But individuals with diabetes insipidus, from my understanding anyway, are constantly thirsty, so they're constantly intaking water, and they're constantly urinating because there's no water reabsorption in this portion. And so normally, since it's impermeable, you can see that ADH is likely there often. The amount of ADH, of course, depends on the amount of osmolarity change difference and that type of thing. So pressure receptors, that will also potentially stimulate ADH as well. But it's mainly the secretion is in those small changes in osmolarity will release some. So it's a decrease of ADH secretion that gives us a little more dilute urine. Okay. All right. Let's see. What time is it? Oh, okay. I want to spend the last 15 minutes or so talking about acid-base balance removal. Let's see, I'm going to go past all this other stuff with kidneys. The acid-base balance is in the first part of your thing, up to about, so I guess there's about 12 pages here. A lot of it is showing you tables in chapter 27 of the different electrolyte balances and regulations. You don't have to worry too much about that. What we really are trying to have you focus on here is regulation of water loss, which this is all involved in, as well as osmolarity regulation, which, you know, here is an example of right in front of us of osmolarity regulation, and acid-base balance, what's happening to have an acid-base balance. So you're going to see some of these. I'll show you one or two really quickly. Better check and see. Oh, yeah, they're here. So you're going to see some of these, a lot of these actually, figures. Oh, so here's hydrogen ion disturbed and what happens. And this is a great one to show because we want to talk about acid-base balance. You're going to ask me where this is, and I'm not going to know because I just threw it up here. But it is figure 27.12, and that is on page 1006. So I would focus your efforts on 1005 to 1010, or 1011 maybe even. Yeah, 1005 to 1011, because the questions that come from here are really focused on this. It has some great information on how you change to blood osmolarity and blood pressure that we talk about in chapter 26. So if you want to look on table 27.5 on page 998. But really the large bulk of what you should be studying starts here with this little figure. And that's on page 1006. The text actually starts with 1005. And just kind of focus on these 6 or 7 pages. So it's been estimated that 70% of all hospitalized patients have some acid-base balance disorder, whether it's a temporary, acute, or a chronic problem. And it is a real problem because our normal body pH is 7 and blood pH is 7.4. If you get up to 7.8, or if you get down to about 6.8, death occurs. So there's not a big range, and actually we see some major things happen as you stepwise go more basic or more acidic. Pretty serious things, including convulsions and a lack of heartbeat. You know, your heart's not beating with inadequate potassium and just calcium. All those things start getting funky, and enzymes themselves stop functioning in a smaller, narrower range as well. And so after, you know, about 7.6 or after about 7.2, lower or higher, depending on what spectrum you're in, you're starting to see some major and potentially permanent damages occur. So it's something that our body has a 3-tiered system in order to regulate. Our moment-to-moment regulation happens in the blood, and here's an example. We'll just say blood pH, let's go down here, blood pH decreases, so you have extra high, your body's becoming more acidic. Buffers in your blood do a moment-to-moment type of reaction. So, you know, within seconds, you see that hydrogen ion can bind up with carbonic acid, bicarbonate to form carbonic acid, and then make water and CO2, which CO2 as a waste product can then be removed from the system. In a minute-to-minute regulation, your lungs can change because we can take large amounts of CO2 and remove them or hold them in. If we hold our breath, we'll hold CO2 in if necessary. In this case, where we have an increase in hydrogen ions, you're going to want to decrease the CO2 so that the hydrogen ions do bind up with bicarbonate. And then finally, something that takes hours and maybe takes a couple days to actually get back to normal is the kidneys. The kidneys can then come in and increase secretion, hydrogen ion secretion, and bicarbonate, or decrease it, as the case may be. So our goal is when your body is more acidic, to get rid of that hydrogen ion, and we do that usually incorporating CO2 in some form, but using the bicarbonate buffer equation. The opposite also occurs. So if you get too basic, you're not getting enough free hydrogen ions, so this becomes imbalanced. you start doing things. You decrease your rate and depth of breathing, so you hold in more CO2. You increase hydrogen ion secretion into the urine and decrease the absorption into the blood. And then ultimately you're trying to get a few more free hydrogen ions in order to get it back, restored back. So this acid-base balance works really well, and it has a couple of different, like I said, tiers to see if it doesn't help. So for example, if you're holding your breath and increasing your POC to PCO2, what happens then is your buffers in your blood will take over and say, okay, combine that with water, which we have lots of water in, and form, hydrogen ions, that type of thing. But then that may get you to acidic. And if that's the case and the buffers are all busy and your lungs, you're still holding your breath, the kidneys can come in, although that takes time. And I don't think you could hold your breath that long. But just as an example, there's a check and balance there to try to help. We consider the cases. So if you have too much free hydrogen ion, you're acidic or have an acidosis. And that's further divided. You either have a metabolic acidosis or respiratory. So either something in your lungs isn't going quite right, or something metabolically is happening, either with the buffer systems or with your kidneys or something like that. The opposite is alkalosis. So an alkalosis condition, once again, either a respiratory or a metabolic condition. There's lots of different examples on how you would get into an acid-base balance disorder. For example, my little girl was really sick with flu-like symptoms. She had a fever, she was achy, she threw up quite a bit, and anything she'd take in, try to give her 7-Up to settle her stomach, or Pedialyte to replace her electrolytes, and she'd throw it up. So she finally just stopped taking in fluid. And we tried. She would have a little bit of water, but she was just so tired of throwing everything up. Well, she got to the point where she didn't have any intake. She wasn't having any output, but her stomach was still cramping, like it was unsettled and obviously unhappy. As we explained it to her, that her tummy was really unhappy. And so she started having major problems. She had a lot of pain, but no, she wasn't getting any release of that she wasn't throwing up anymore. And we took her into the Instacare because it happened to be Friday the 3rd, and they put her into the ER, said she was severely dehydrated, which we kind of suspected she might be. She was getting a little lethargic and a little lightheaded, and gave her some fluids, and she was fine. We got to go home that late afternoon, and so she's fine now. She's back to her normal self. But when they did her blood work, it shows that she was in a little bit of an altered state because she had lost so many electrolytes. So she was having a hard time with that. And so she was a little dehydrated. And so that was a metabolic. And in this case, she was a little bit acidic because she was combining a lot of her CO2 and water and making acids. But someone with smoking and nicotine damage would also be a metabolic acidosis or a respiratory acidosis, depending on how severe that damage is. For instance, like emphysema, because emphysema is a lung disease, would be considered respiratory. Anyway, so there's lots of different, if you're on a high protein diet, there's another one where you can put yourself into a metabolic state of imbalance. So there's lots of reasons that could happen. Often they're what we call acute. They're not chronic symptoms like emphysema would be a chronic acid-base balance disorder. But you'll see on page 1010 and page 1011, there's a clinical impact and there's a table about acidosis and alkalosis and what's happening in your body. And you need to really kind of study those because the questions that you'll have result from those 2 pages and the couple of pages just kind of explaining the buffer systems. So you need to understand that all 3 systems are involved: the cardiovascular in circulation, the respiratory, and the urinary system are all involved in acid-base balance. And that specifically when you're out of balance, so when you have acidosis or alkalosis, that you need to know what's happening and what your body may do to try to compensate for that. Anyway, so just know that it tells you what happens with what happens and what you'll learn. And your book's not really clear on this, so I'm going to just give you a little hint. What you'll learn is it's the opposite that's your compensatory mechanism to compensate. So, for example, if you have a respiratory acidosis problem, like emphysema, then you're going to have a metabolic alkalosis. Your body's going to induce that to try to balance it. So it's like a seesaw. So if you're on one and you want to try to balance it, you're going to do the opposite effect. So just remember that when you get those quiz questions and those exam questions, that whatever the problem is, if you get the problem correct, say metabolic alkalosis, then your body is going to go into a respiratory acidotic state in order to compensate. So if you have metabolic alkalosis is what I just said, so a high pH, your body is going to reduce your respiration rate. You're going to hold on to more CO2 to try to correct that. You're trying to get yourself into a little bit of an acidotic state so you can correct it and get enough free hydrogen ions in there. So that's the main point as far as trying to, not that. No, go away. Trying to understand that is, remember, it's the opposite. Your body tries to do the opposite. So it tries to hold on to. Either hold on to CO2 or blow off CO2 or secrete hydrogen or secrete or decrease reabsorption of bicarbonates. In other words, it's trying to remove it from your blood so that it ends up in the urine. So think of it in that way. If we're trying to get stuff into the urine, it's, hydrogen ion into the urine, or we're going to have less hydrogen ions in the urine itself. Okay, so just know I was looking for, let me see if I have one more thing to show you. This might help. Look here. This is a really old picture. I'm hoping this is the right picture. It might take too long. It was taking forever. Some of these that are scans are, this is a scan picture. I'm going to see if I can go to it really fast in my folders. Since it's still waiting to come up. Watch it be the wrong one. Oh, yeah, so this, that's a kidney disease. We don't need that one. Cancel. Try this one. This is just a little JPEG of, I didn't say that folder, of acid-base disorders. And it's going to have a little more information than you probably want on it. But I'll start talking about it if I can get back in there. Okay. So this shows the defect, the compensation, and actually, ultimately, this is, like I said, a little more in depth than you need to know, but kind of the calculations of how to figure out what it should be. If you go into nursing or if you go into healthcare in a hospital situation, you'll probably learn about this in a different mannerism, but I hope you can see that okay. But if you have a metabolic acidosis, you have too little bicarbonate, this is your compensation. You're going to hyperventilate to blow off a bunch of CO2, and this opposite effect occurs if you have metabolic alkalosis. The first thing you're going to do is try to hold on to the CO2. If you have respiratory, and that means you have too much. In respiratory, it's the partial pressure of CO2 that is too much. And so your compensation is to try to increase your bicarbonate so you can make that equation go from water to CO2 and then get more free hydrogen ions metabolically. Same with chronics, are going to be a little different. We're going to try to, compensation will be in the kidneys because your buffers aren't going to be able to handle it. And that's the difference between acute and chronic when it comes to respiratory problems. Chronic, the kidneys have to do the compensation because your buffers can't handle that imbalance. Most of the time, metabolically, the compensation is respiratory. It can handle it okay. Respiratory compensation. If it's acute, the buffer systems can handle it. If it's chronic, the kidneys kind of have to come into play. You never get into a full, complete balance, pH balance, without some effect or compensation from the kidneys. And that does take hours and sometimes takes days. And so ultimately your body's good at handling those slight fluctuations without having to go to that step. So once again, I know that's kind of an older scan and it has a little bit extra information, more like a 460 level, but this part here is still helpful if you want to kind of look it over and read that clinical impact and kind of look at that and say, oh yeah, that makes sense, that if I have this problem, my body's going to do this. Okay. Are there any last minute questions? Focus on acidosis, focus on what happens through the nephron. Okay, well, have a great night. I hope that was somewhat helpful for you. Next week, lecture is going to be a little bit earlier in the day on Tuesday, and I will announce that as it comes up. But our last lecture, and it's also a little longer. There is, Amelia, if you go into the notes from instructor, it says lectures from Sister Foster, and then all the links are there. So notes from instructor, if you go down to lectures from Sister Foster, including class meeting, I think it says, you're going to find them all there. And this one will be posted later tonight. All right, have a good night. Thank you for coming, and hopefully I'll see you next week. Like I said, it'll be a different time. I don't know if it'll work for you, but if not, then send in your questions. I'll make sure to answer them. All right, thank you. Have a great night. I'm going to go ahead and stop the recording.